So it is my distinct pleasure to introduce Kapil. He is a postdoctoral associate in Rohit Singh's lab here at Duke University, and his research centers on developing and applying protein representations to characterize protein structures, functions, and binding interactions. And today he's going to be telling us about one of his newest preprints, Raygun. So with that, Kapil, we're very excited to welcome you, and please take it away.
Yeah, can anybody hear me? Yeah. So, hello everyone. I'm Kapil Devkota. I'm a postdoctoral associate at Duke University, working in Rohit Singh's lab. And this talk is going to be about a new paradigm of protein design called template-based design. And I'm going to explain it using a new protein design tool created by our lab called Raygun. So, this talk is going to span a lot of things. First, we are going to talk briefly about the existing de novo approaches and what the challenges they face in their usage in basic science and translational research. Then we are going to talk about what template-based design is, how Raygun incorporates it, and the applications that are now possible. Then we are going to talk about the Raygun model architecture. And finally, I'm going to talk about some results obtained from computational and biological evaluations, computational and experimental evaluations. Now on to the first part. In the domain of structural biology, the development of methods like AlphaFold 2, OmegaFold, and the recent new version AlphaFold 3 have made the structural inference of rigid globular proteins essentially a solved problem. That is why the main focus has moved on to de novo protein design. So instead of inferring structure from sequence, you are given partial information which can be part of a structural motif, part of a sequence, and probably just a function tag, and the goal is to fill in the gaps. So either create a scaffolding for the structural motif or complete the sequence given some of its part, and so on. The most popular example of de novo design method is a method called RF diffusion. It's a diffusion based method which can take in different type of structural motif, and the goal is to fill in the rest, and it can do different kind of constraint and unconstrained design, and in the constraint design it takes various type of structural motif and creates a viable scaffolding. So the design challenges and biological possibilities associated with the de novo design methods make them a very interesting and challenging problems, but there are different issues that needs to be addressed. One major issue that de novo design faces is designing proteins with particular function. We know that de novo methods can be used to produce structurally and sequentially viable proteins, but function is an entirely different thing. It has a qualitative aspect to it, and there is no general solution that maps sequence to function or even structure to function. Therefore, no matter how good of a structure you can create with de novo design, viability does not necessarily entail functionality. That's why it's important to remember this, as we will come to this point later in our experimental validations, where we found that a sequence with the best structural property often failed to recapture the desired functions that we wanted. And the second limitations that de novo design usually have is that in the biological settings, optimization is not done from scratch. Usually have a protein at hand, and the goal is to optimize it. For example, you have proteins like fluorescent proteins, which is a very important class of protein used for detecting localization and protein activity. For this class of protein, biologists are more interested in different optimization problems like, can you make this protein fluoresce more? Can you make it more functional? Can you increase the spectrum of its emission? Another question can be, can you miniaturize the protein and make it less disruptive so that it can be attached to smaller proteins? And there has also been exhaustive work done on making the protein circularly permuted, which basically means bringing the C and N terminus of the fluorescent protein close so that it satisfies some properties necessary for creating fusion proteins. So these are proteins with known functions, and they want to optimize the functions so that it can add or enrich the existing function. There are other problems that biologists are interested in, like the very important class of proteins called Cas9, which is very important protein and used for DNA editing. For this kind of protein, they actually this protein is very big, so it cannot be directly passed into the organism. It has to be created endogenously using transgenic mouse line. So can we make the sp-Cas9 and protein that are large like this sp-Cas9 with greater than 1100 amino acid? Can we shrink it so that it can fit inside usual delivery vectors like viral delivery vectors? So being able to find a shrunk version of Cas9 is has a very big consequences, as it effectively mitigates the need for transcending mouse line and significantly simplifies the biological pipelines of experimentation. So this kind of biological design problems, it's unclear how de novo methods can be used. So our goal is to take the first step towards computationally addressing these important problems, and in the same time generalize it for a broader range of applications. Now we move on to the second part where we describe what template guided design is. So the template guided design should take existing sequence as a template, should any sequence as a template, give wide functionality to miniaturize, magnify, and modify the template protein while retaining function. The third thing it requires no domain information to operate. So it should require no domain information to modify the protein. And it should potentially give users control over the sequence diversity and target length while preserving template function in some way, right? So existing methods of template guided design exist but are very primitive, and they are limited to few substitutions. If you want to do more than one substitution, that poses immense combinatorial challenge. So in order to overcome this combinatorial hurdle, you require some domain knowledge so you can guide through this combinatorial problem. Also, uh, requires domain knowledge on where to edit, so to solve this combinatorial problem. Also, it cannot produce, and it cannot model higher order properties like epistasis, which basically means mutation at two sites is not equivalent to the combination of their separate effect. And obviously it cannot handle insertions and deletions. So our solution to this template design problem is a fully functional template guided design tool called Raygun. The novelty of Raygun is that it can be used to miniaturize and expand a protein, and it takes in any template protein greater than 50 amino acid. It does not require any domain knowledge for sampling and prediction, and it gives you full access over the sequence diversity. You can miniaturize, magnify, and modify while retaining function. And finally, it gives you full control over the target length and the sequence diversity. Now, we found in our evaluation that the output sequence obtained through Raygun likely folds very well. It's very likely to have a very similar sequence structure, and very high rate in basic functional validation. So this brings up very interesting possibilities with Raygun. For example, we can use Raygun to remove or adding loop in the sequence, and with loops we can add functional groups within this loop. We can use it to shrink bring proteins so that it can be fitted inside a standard delivery mechanisms like AAV. It can be used to make smaller sensors so that there is no interference with the actual reactions happening. So to make the sensors less disruptive. And finally, it can also be used to refine a function of a sequence, right? So how does Raygun incorporates template guided design in its internal architecture? We are going to explain that in this section. So Raygun is basically an autoencoder model with one novelty. The novelty here is that the encoder part can shrink any variable length protein into a fixed dimensional space. So you have variable length sequence here. You can take any length input, and the encoder layer converts the variable length embedding obtained through ESM2 to a fixed length representation space. And that is done through two simple and parameter-free layers called reduction and repetition layers. And the fixed length representation is transferred to the variable length representation using any target length we desire using the decoder layer, right? So in order for this basically model is a embedding to embedding model. So you have a ESM2 embedding, you project it to a fixed length representation space using encoder, and you project back to the variable length space to using the decoder. So this embedding and to embedding model would only be useful if you can convert the embedding back to the sequence space. So we need embedding to token decoder in order to make this model useful for sampling and evaluations. Turns out we can easily do that by adding a simple prediction head on the ESM2 embeddings. So we created a simple prediction head that is composed of some linear layers and a transformer encoder layer. And then we ran it on SwissProt. We trained it from SwissProt data, and we got greater than 0.99 accuracy. So we use this to convert the embedding to embedding Raygun model to a sequence model, where you can take in sequence and you can sample the model and return the sequence output. So let's talk about the layers that are used for fixed to variable and variable to fixed length representations. These are done using two length transforming layers. One is the reduction layer that transforms variable length protein embedding to a fixed length encoder space. Then the other is the repetition layer that does the opposite. Now we have decided to make these two layers complementary and parameter free. And we add complexities before and after these length transforming layers. But within this, we wanted to make the reduction and repetition process as simple as possible. So how does reduction layer does this? The essential thing we do in the reduction layer is we have a variable length representation of ESM2 with length equals to L. What it does is it divides the protein representation into K contiguous block, and in our case, K equals to 50. And then reduce each block using a simple averaging operations. The averaged output within each block is then concatenated again to get the final output. So we can also conveniently do a standard deviation within each block to get the standard deviation and return that as well. So this reduction process is very simple and this does not require any parameter, but it adds a significant distributional properties to the end result. If each blocks are assumed to be loosely dependent, each vectors in the blocks are assumed to be loosely dependent, and the window size of each block is large enough, then the distribution of the output can be estimated as a Gaussian through central limit theorem. So you can estimate the end result of the reduction process as a simple Gaussian, and we can sample from this Gaussian using simple mean and variance operation. So just by finding out the mean and standard deviation values, we can effectively sample the output of the reduction layers. This is a very interesting property and very useful because this property makes the single shot sampling feasible. Because unlike diffusion models, which requires many iterations, sometimes 100 or 200, to move from sampling distribution to the target distribution, we can directly estimate the Gaussian of the Gaussian on the reduction layers. And we can just simply sample from this reduction layer. So, and we can just use decoder to transfer the sampled regions back to the protein representation space. So this makes our sampling and inference much orders of magnitude faster than conventional diffusion-based models. And the things are not as straightforward as it look. It is impossible to have the same window size for all proteins because theoretically only 1 in 50 proteins have length that is multiple of 50. For example, in the SP-Cas9 case, if you set the window size to 27, there is still going to be a remainder of 18. Where are we going to place these remainder terms while ensuring the overall process is differentiable? That's the main question we faced. So the first thing we did was attach the remainders obtained at the start and end blocks. That one is not consistent across block because you have start and end blocks that are of different lengths, and it caused most of the protein edits to go at the start and the end layers. So the editing bias was at the extremity of the protein sequence. So that was not really good. So the second thing we did was, why not divide the sequence into three regions: the start, middle, and end regions? And the start and the end blocks would have window plus one size. So originally the window size was 27. The start and end blocks will have 27 plus one size, and the middle part will have the default window size. So we are spreading the remainder over a large span. And this makes things more streamlined, consistent across the block, and there is no bias at particular sites on where Raygun is going to edit. So this makes things much more balanced. So this remainder is spread over the start and the end blocks, right? So now for the reduction layer, it works exactly complementary to the repetition layer. So repetition layer works exactly complementary to the reduction layer. You have a fixed length representations and you have a target distribution, you have a target length at hand. What you do is, you based on the target size, like in the reduction layer, you divide it into three blocks: start, middle, and end. And then we repeat each vectors by the window size. Like in the reduction layer, the window size at the start and the end blocks is going to be one more than the middle blocks, and concatenating them all together gives us the final results. So the reduction and repetition layer does not require any parameter and are very simple operations of converting fixed length to variable length and variable length to fixed length representations in a differentiable manner. So we can pass the gradient backward from these layers to the actual parameterized layers. So in the overall architecture, how are these reduction and repetition layer used? So this is the encoder part, which uses the reduction layer to do variable to fixed length representation. The reduction layers are positioned between the TE blocks, which is basically transformer encoder layers with added convolutional filters. So transformer layers and convolutional filter is added so that both long-ranging and short local relationships are mixed properly before and after reduction. And the decoder portion has the repetition layers interleaved between the two transformer blocks. Notice that the decoder takes additional target length as one of its input. So you can use decoder to transfer the fixed line representation to any length you want. Also notice that sampling only happens in the reduction layers in the encoder. We made this reduction layer do this simple averaging so that the end distribution can be estimated as a Gaussian. We exploit that here and sample only at this location. And although the output distribution of the reduction layer, being a simple Gaussian, is a very simple and naive, the machinery that we have added before and after the reduction layer adds enough complexity to potentially allow the distributional sample over the whole ESM2 embedding space. So even though the output distribution of reduction layer is simple, we have added the parameterized layers with higher complexity and higher parameters so that it can incorporate all the possible ESM2 embedding space. So how did we train the Raygun model? We trained Raygun as an autoencoder model where we passed the ESM2 embeddings and expected a perfect reconstruction by the decoder. We did not train it explicitly as a variational autoencoder because we thought that the sampling mechanism through the reduction layer we generated and we designed would be robust by design, so we did not need it to train it as a variational autoencoder. And we did not want the decoder to be burdened with two different tasks of one, removing noise, and also perfectly decoding the fixed length embedding. So in order to make the perfect decoder, we sampled from uniref proteins of length between 100 and 1000. We also ensured that we took all length between 200 and 200 and 1000 into account so that the decoder is more robust to length variabilities. And we use three training losses. The first loss is the reconstruction loss. is basically ensures that there is a perfect reconstruction of ESM to embedding at the input and output length, at the same input and output length. We have a cross entropy loss, which uses the ESM2 token decoder and checks if the logic obtained matches the input sequence. And finally, we have the replication loss, which checks if the fixed length embedding has a kind of a circular property, i.e., suppose a same embedding is used to produce two different embeddings of two different lengths, L1 and L2. When these two different variable length embeddings are again passed to the encoder, they should produce the same fixed length representations. So this kind of circularity condition is added as a added regularization loss to ensure that the model is trained well. And so that does the training part. For the sampling portion, recall that our one-shot sampling from the reduction layer has made the models orders of magnitude faster. Actually, we can do more than 6 samples per second using simple A6000 GPUs. So we wanted to exploit that. So we wanted to exploit this high throughput ability of sample generation through Raygun. So what we did was we exploited this by adding filtering tools to select among the candidates that are produced from Raygun. We use we could cascade filtering tools one after another. These tools can be structural, functional, methods like jackhammer and more. But one primary filtering that we added was a single shot approximation of pseudolog likelihood by Nadav et al. To select for fit protein candidates. We found that this is very fast and this works pretty well. That's why that is used as a primary filtering tool in every sampling problem that we did using Raygun. And we also found that PLL is kind of length dependent, and we applied measures to correct for that, and that we describe in the paper. So now to the evaluation part. How did we evaluate our model? The first thing we did was we selected a small protein like hemoglobin monomer, which has around 147 residues. And then we tried to shrink and expand it across a reasonable length spectrum, 20% smaller and 20% larger. We see that it actually preserves the structure quite well for that spectrum because the structure is kind of preserved over the entire length. We also show the MSA and the red highlighted sequence here represents the template, original template. Notice that we do not provide any additional knowledge. We do not provide any domain information, but Raygun is finding out which region is important, which regions can be removed, and which needs to be substituted. Just in a way, a biologist with domain knowledge carry out such editing operations. So we can do what biologists do in editing proteins without the requirement of any domain information, which makes this a very important and interesting tool. We also did length modification for different size proteins. Notice for that large proteins like mTOR, we were able to shrink significantly more. And that makes sense because for larger protein, the window size is larger. So our assumption of Gaussian distribution as the end result of any reduction operations becomes more accurate. So potentially sampling larger protein gives us better candidates than smaller proteins. And we also wanted to see if there is a bias in Raygun, i.e. many designs are known to prioritize helices. And we wanted to see if actually Raygun also have that kind of bias. So what we did was we took 4 PFAM domains, alpha only, beta only, alpha plus beta, and alpha by beta corresponding to these scope classes. And for each classes, taking alpha only domain as an example, for explaining our experimental design, what we did was we took 5 proteins belonging to that domain. So for this alpha only domain, we took 5 proteins. These 5 proteins were selected to cover the entire length spectrum. So smallest to largest, they cover the entire length spectrum of the PFAM domain. We took the protein with the median length and used it to decide the length spectrum over which we are going to perform the stretching and shrinking. So this is the median length protein. We used it to produce length spectrum, minimum length 209, maximum length twice at 836, and that is divided into 20 bins. And for each 20 bins, we generate 5 candidates from each protein. So in total, it produced 25 candidates per bucket. And the size of the circle basically represents, out of the candidates, how many retained their original PFAM domain. So if there is no circle here, that means that none of the candidates we generated actually retained their PFAM domain. And the size of the circle basically implies what percentage actually retained their domain. And we also computed statistics per bin. And finally, we showed some of the examples. So for alpha only domain, we found out that 222 out of 500 domains were actually preserved. For beta only, there seems to be a certain threshold beyond which if you sample and if you reduce the length, it does not work. The PFAM domain is not preserved. But beyond that threshold, it actually has a wide range. Like for this protein, it has a pretty high shrinking and stretching capabilities. For alpha by beta 282, more than 50% of the candidates we generated had their domain preserved across a wide range of length spectrum. And for alpha plus beta, it was the worst performing. Only 187 out of 500 had the domain preserved. But it also looks kind of good because there are some candidates which actually were more amenable to shrinking and stretching. So on the whole, we found out that there is no visible and noticeable bias in Raygun in doing protein editing, and it can be used to shrink and expand and preserve the required PFAM domain for a wide variety of secondary structures. In general, we found out that the function is largely retained if we did length modification by plus minus 10%, although for some proteins, we can modify it by a even larger margin, more than 50% deletion. We can also control sequence diversity by changing the noise parameter. We found out that setting the noise ratio to between 2 points below 2.2 and above 0.5 gives a good balance of sequence diversity and good structural alignment. And we also did many other computational experiments, and you can see that in the paper. And final section is experimental validations. We validated our model using 3 assays. The first assay was trying to create smaller fluorescent proteins. So fluorescent proteins are important class of biosensors, and around 25% of human proteins are smaller than fluorescent proteins. And our goal was to find the smallest fluorescent proteins that were cofactor free. So we did use this pipeline. We exploited the high throughputness of Raygun to generate 140,000 candidates for eGFP and mCherry. And these are basically, eGFP is green fluorescent protein, mCherry is red fluorescent protein. And we use single shot PLL filtering that we described before to remove 90% of the candidates. And then we used jackhammer and another brightest tool that we created using the FPbase data to figure out which of the candidates actually preserved their domain and predicted, sorted them based on the inferred brightness. And finally, we used AlphaFold based PLATT scores to do final filtering and generated 8 candidates. We like to remark that people have actually done research on fitness landscape of fluorescent proteins. And through the directed evolution and through DMS, they have found that the emission landscape of fluorescent proteins are quite finicky. And 5-point mutations are almost always going to kill the fluorescence. So we are trying to do more. We wanted to see if larger number of mutations, addition of more insertions and deletions, would still cause the fluorescent to be retained. So these are the 8 candidates generated, 4 from eGFP and 4 for mCherry. There are some candidates like candidates 5 and 8 that had pretty bad PLDTT and TM score values against the original mCherry. So in fact, except we ate that the candidate, this did not even have the same chromophore as mCherry. So we did not force the model to preserve the chromophore region, and we did not add any domain specific information. And we selected these candidates because within this length spectrum, we wanted to do experimental validation over a long range spectrum. And within this length spectrum, these are the best performing sequences that we obtained. And we just bit the bullet and try to do experimental validations on these 8 candidates. also including these very low PLDT candidates. What we found out that the lowest structural integrity, the candidates with the lowest in structural integrity actually fluoresced. Even 199 amino acid that did not preserve the chromophore had fluorescence in it. So just to be sure, what we did was we added a nuclear localization tag, and we wanted to see if it actually localizes to the nucleus. And we found out that out of 8 candidates, only one candidate did not show fluorescence, and all of the candidates, even the candidates that did not have any uh, chromophore, the original chromophore, actually fluoresce, right? So these are the candidates that PLDT showed to have very bad alignment and very bad scores, but still it produced quite well. This indicates that although PLDT is a good measure for protein rigidity and viability, like we said earlier, viability might not mean the same as functionality, and it might not be the perfect metric that captures the function specific fitness and folding efficacy of proteins. Another assay we did was on two important synthesized biotin ligases called turbo-ID and ultra-ID. These proteins are very important. They are able to biotinylate themselves and the surrounding proteins are very useful in PPA predictions. And one thing to note is that ultra-ID was created from turbo-ID by removing this HTS DNA binding domain from turbo-ID. We use the similar filtering pipeline while replacing brightness with thermostability to produce more thermally stable candidates. And we generated 20 candidates. Remember that BRA 1 and 2 are wild types. So actually we produced BRA 3 to 20, which are basically 18 candidates. So these are the different PLDT and TM scores we obtained from our methods. We found we have more turbo ID candidates than ultra ID. And for candidate number 20, since we told earlier that ultra ID is constructed from turbo ID, we wanted to see if Raygun can actually simulate that. So what we did was we had turbo ID, we asked it to reduce the length of turbo ID by 50%. And we just, the original length was 320. We tried to reduce the length by 50%, and this produced this protein. And we found out that the Raygun generated candidate actually removed the STS domain, and it looks kind of similar to ultra ID, and that was kind of very interesting. And we also tried to experimentally validate it, even though it was a long shot. And turns out of the 20 candidates 18 candidates we selected, 7 actually folded, even the candidates we shrank by more than 50%. Even better, 6 out of 7 candidate was strong enough, had retained functional property to biotinylate itself. It was not able to biotinylate other candidates, but it retained the property of biotinylating itself. So even though the property is dim after the editing process, the functionality is still retained and it is able to fold, which is a pretty interesting result. And I think we should probably have to do more directed evolution to make the functionality go even stronger. And the last evaluations that we are going to do is we are going to do an assay to shrink cast time. So the target length, we want to reduce it to less than 1100 so that it can actually fit inside AAV delivery mechanism. And we are going to run validations on 10 candidates. This is one of the AlphaFold inference of one of the candidates that we produced. And it looks quite well. It has a good AlphaFold PLDT scores, and it also actually shows double strand break, which is optimistic. And we hope that it will also do it in real experimental assay. So in conclusion, template-based design opens up new possibilities. Raygun can incorporate this approach to generate functionally and structurally similar candidates. Evaluations that we did were done on fluorescent proteins, turbo ID, ultra ID, and Cas9. And for future works, we want to do many stuff one of the stuff is adding in silico directed evolution to make the generated Raygun candidates more functional. Finally, the code is available here. We would want people to look into the code, give us suggestions to make it more accessible to computational people and biologists alike. And finally like to thank Sorting Lab and my PI. In Sorting Labs, I want to thank Scott, Daiji, and Zoe for their help in experimental validations. And thanks for your time, and we are open to collaborations. Thank you.
Thank you so much. People can feel free to unmute themselves if they would like to, or they can also put the questions in the chat. So I'll just give you guys a couple minutes if you want to type anything up, but also please feel free to unmute.
Yeah, any questions, suggestions?
I guess getting started, I do have a question. So
Sure.
I am wondering if you were wanting to work with some larger enzymes. There are some enzymes that like oligomerize. If you think that you would actually be able to shrink those and still retain those protein-protein interactions that are like vital for that oligomerization. Have you guys thought about that and thought about if Raygun would work well for that if you wanted to shrink the monomeric form of the protein but still be able to see it oligomerize?
I think we actually want to do that. We want, like, oligomerization would involve us trying to preserve the structure and also preserve the PPI, right? Interactions with itself. And I think that is a more difficult problem to address. And I think we would be interested in doing similar stuff actually. Yeah, but that is definitely a more difficult problem to solve. What we can do is we can use Raygun to shrink the candidates and see if it can oligomerize, right? So yeah, there's a good experimental pipeline that can be used for validation of that kind of experiments.
Hello,
Yeah.
Kapil.
Hey,
Hi, this is Arkady. Thank you for this great presentation.
thank you.
Could you go into a bit more detail how your length, like your length request that you put into the decoder, that's basically an integer that you pass, right?
Yeah.
How does that get attached or appended to your decoder? Or how does it exactly get used in the decoding process?
So you want to understand how this attachment happens here?
Yeah, yeah, the length and
So yeah, so transformer layers are length invariant, right? You can provide any length to transformer and it will produce the same output at the end, right? So this basically, we try to imagine a unit-like architecture where you have multiple repetition copies, right? Copies from one that goes straight here, fixed length representation converted to variable length representation here, and those are uh, we add more complexity here. Like it's kind of like a UMAP like net that we wanted to design. So we put the fixed length representation here, it will produce a fixed length representation, and then we will repeat it. And then we'll add further more complexity. We do this multiple iterations. Actually, there are 10 layers here. So we do it 10 times and we add more complexity here. So there are multiple replicates forming, and finally all the replicates are concatenated and projected to the required dimensions to produce the final variable length representations. So the first path is the simplest path. We are just taking the fixed length representation and repeating it and project and applying a simple transformer on it to produce one replicate. And then we are adding complexity by appending multiple transformer blocks one after another. And finally, all of these are concatenated and then projected to the final layer. Does that make sense?
Yeah, thank you.
Yeah.
Awesome. Well, if there's no other questions, I just want to thank you again, Kapil. Also, thank you to your professor, who I believe was in attendance at the seminar today. This work is amazing, and we can't wait to see like what's to come of it. So thank you again for giving your talk today.
Thank you. Thank you. Thank you for letting me present.
Awesome. Yeah. And then for everyone else in the community, we'll see you guys in a couple weeks for our next seminar. Thank you.